As we wrap up cell transport, the last thing we have to talk about is active transport. This is going to be the exact opposite of passive transport in that we are going from a low concentration to a high concentration. So we're actually going against the concentration gradient. If you remember our ramp, we're now going up the ramp, or we're going upstream. If you're paddling upstream, it takes a lot of energy to go a certain distance. Now with active transport, we are specifically looking at kind of two different types of functions. We have what's called your protein pumps, as well as vesicle transport. We're focusing on larger molecules or basically ions to kind of make this happen. But the key thing in making active transport is we need that ATP energy to go from your low to high concentration. So our first type is our ion pumps. We are transporting the ions across the membrane against the gradient with that ATP, going from that low to high concentration. The pump that we're going to talk about the most, or that you'll hear about the most, is the sodium-potassium pump. Sodium-potassium pump plays a key role in the electrical signals among our cells. And as we see here, we have more sodium ions outside of the cell. We have less inside of the cell. However, we will always see sodium leave from inside the cell to outside. In contrast, we have more potassium inside the cell. However, we see more potassium come inside. In order for sodium to leave and potassium to come in, we need ATP to kind of power this process. Sodium-potassium pump. So going up the gradient or against the gradient, low to high concentration. Remember, we need an apples-to-apples comparison here. What is the concentration of sodium inside and outside the cell? What is the concentration of potassium inside and outside the cell? Our second type of active transport is our bulk transport, when we are bringing in large objects or large molecules. Endocytosis, endo means enter. So as things are coming into the cell, we have a vesicle that forms. Kind of forms like a little pocket, and then it pinches off to form a vesicle, and the vesicle will kind of take it where it needs to go. Typically, your lysosome will break down your vesicle to release the contents into the cell. So we lose a little bit of cell membrane every time this happens because the vesicle is made from the phospholipid bilayer. The opposite of this is exocytosis. We kind of already talked about this with the first part of this unit with the endomembrane system, how our secretory vesicle goes to the cell membrane. Well, if we're carrying our proteins to exit out of the cell, that vesicle is going to fuse with the cell membrane in the process of what's called exocytosis. Exo means exit. So we want those contents to leave the cell. So our second process that we would call bulk transfer or bulk transport. And when this happens, the cell membrane gets a little bit bigger since the vesicle is fusing with it. But everything kind of evens out because in endocytosis we're losing a little bit because it's entering the cell. Exocytosis, our cell membrane is gaining a little bit since the vesicle is fusing with it. So which type of cytosis would increase the size of the cell membrane? It'll be exocytosis because the vesicle is fusing with the cell membrane, making it a little bit bigger. Endocytosis would decrease the size of the membrane since we're pinching a little bit of it off to make the vesicle entering the cell. quick review.
Recall that there are two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy. Active transport also moves from an area of low concentration to an area of high concentration or up the concentration gradient. This is the opposite of passive transport, and because it's more difficult, it needs the energy molecule called ATP, which stands for adenosine triphosphate, because there's an adenosine molecule and 3 phosphate molecules. There are 3 types of active transport: membrane pumps, endocytosis, and exocytosis. Let's look at membrane pumps first. Membrane pumps are just carrier proteins that move substances from areas of low concentration to areas of high concentration. Let's look at the sodium-potassium pump to see how it works. Outside the cell, there's a high concentration of sodium ion, or Na+. The brackets represent the word concentration. Inside the cell, there's a high concentration of potassium ion, or K+. The cell has ion channels that will allow these ions to diffuse and reach equilibrium, but the cell doesn't actually want that. It wants to keep a difference in concentration. So the sodium-potassium pump will force more sodium out and more potassium in all the time. Let's see how this works. First, 3 sodium ions from the cytosol bind to the protein, and a phosphate group from ATP breaks off in the carrier protein. This provides energy for the shape of the protein to change, and the sodium ions can be released into the outside of the cell. Now 2 potassium ions will enter the pump, which causes the phosphate to break off and return the pump to its original shape and release the potassium into the cytosol. Again, sodium will diffuse into the cell on its own, and potassium will diffuse out. The pump forces the sodium to be outside the cell and the potassium to be inside the cell. Another way that cells do active transport is through vesicle movement, either endocytosis or exocytosis. Endocytosis brings items into the cell, and exocytosis pushes them out of the cell. Endocytosis is the process by which cells ingest external fluid, macromolecules, or other large particles, even other cells. We can be more specific about what materials undergo endocytosis. Phagocytosis is where large particles or whole cells are ingested, and pinocytosis is when solutes or fluids are ingested. You can think of phagocytosis as cell eating and pinocytosis as cell drinking. Here you can see the way it works. First, the cell membrane begins to pinch around the substances, and eventually it forms a vesicle, which can carry the substance to any part of the cell. Exocytosis is basically the opposite of endocytosis. It's a process by which a substance is released from a cell through a vesicle that transports it to the cell surface and fuses with the cell membrane. A vesicle can merge with the cell membrane because they're both made of phospholipids. The layers press into each other, and the phospholipids rearrange a little so that they can open up their contents to the outside of the cell. Again, this requires energy, so it's classified as one of the types of active transport. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and follow me on Twitter at SciencePet.
Okay, so a little quick review. So on the bottom of page 17 in your packet, we have 6 different transports. Remember, osmosis is passive. That is the diffusion of water. No energy needed because we go from high to low concentration. Endocytosis is one of our vesicle transports. We need energy to kind of move big materials into the cell. So since we just require energy, it is active. Ion channels. We talked about this with facilitated diffusion. An example is an ion channel. So this is passive. Facilitated diffusion is passive, no energy needed. Exocytosis is our other vesicle transport, whenever you're moving big things in the cell or into the cell or out of the cell, it requires energy. So active transport, and then our sodium-potassium pump is our other type of active transport in regards to moving sodium and potassium and kind of making sure that difference in concentration is made intact. So since it is active, yes, energy is required. So the big takeaway today is understanding active transport. We do have our sodium-potassium pump. We are going against the gradient with that. We also have our vesicle transports where we are bringing in or getting rid of large molecules. We briefly kind of introduce this with our endomembrane system. All right, that marks the end of transport. If you have any questions, please ask. Thanks for watching.